So we're continuing our discussion of the lac operon, this time getting into a little more specifics about the genes and the products of the genes involved. There's some terminology here, basically the names for these regions. These are the names that they were named as they were discovered. So they get this LAC, lac prefix, and then they're basically just identified as important gene regions. And over time, as the scientists were figuring out how this all works, they would piece these together and make sense of them. So they're not necessarily a lot of order to how they're named. You can see A, Z, and Y are the structural genes in there. Just sort of placeholder names, unique names that later became associated with the functions of these various places. So what I want to do by telling you that is that I don't want you to learn these things as very important names. Rather, I'd like you to remember what's important about the process of gene regulation here. So I did just want to tell you what's going on with the gene names. They're not all regions that are protein coding, but they'll sometimes have other functions. So start in with this P sub I region. This is promoter for this other region called lac-I. So the promoter for the lac-I gene, P sub I is its promoter. So it has its own promoter, it has its own protein coding region in red there. And then by transcribing and translating the lac-I gene, you get this repressor protein. So again, as I mentioned before, the protein is an enzyme and has to have its own DNA instructions. So that's where the repressor comes from. It doesn't have to be located anywhere near the lac operon, as the important molecule that does the effecting is this enzyme here that can go really anywhere else in the genome. So it could be made near to the lac operon, or it could be made far away, and this repressor protein was going to find its way to the promoter either way. So sometimes I put a little like jagged zigzag in here to indicate that these aren't necessarily neighbor regions in the genome. So once that goes, the repressor attaches to a portion of the promoter. This terminology I find a little bit more detailed than we really need. They have the promoter, which is lac-P, and it's in the same color as that other promoter. And then there's a subset of the promoter called lac-O, which they call the operator. And that is specifically where the repressor molecule attaches to the DNA. So it's really part of the promoter, but it's an important sequence of DNA nucleotides present in the promoter that serve as this operator, the physical attachment point for the repressor. So you can think of the operator as just being part of the promoter. Again, I'm going to try not to grill you on the fine details of the naming system here. So shown here, as the title indicates, this is the lac operon in its off position. If you remember, this is an inducible system, so it's usually off in the absence of any signal molecule. This is off, the repressor is made, and the repressor attaches to the promoter region. So this, in the absence of whatever fits here in the circle portion of the repressor, as long as that molecule is absent, this whole thing is just going to be shut off forever. So it's kind of like the spigot on your water faucet outside. It's usually off, even though the water is pressurized and ready to come out anytime. Having it off really shuts that down, and nothing ever happens of this for transcription until the conditions are right. So here we have the same situation. The DNA is the same as before, the positions are all the same, and we just see a different level of activity. So the repressor molecule is still made, but this time the circle part of the repressor molecule is able to interact with lactose. I'm going to use lactose in place of allolactose. Again, that's a level of detail I think you don't need to know. So lactose attaches to the repressor, and that prevents it from repressing. So this is a minus on the minus, and that enables RNA polymerase to do transcription. So this is a situation where RNA polymerase can do the transcription by itself as long as there's nobody in the way. So this repressor getting out of the way allows transcription to happen, transcribes through all three of these genes as part of this operon. So one RNA for three genes, each of them gets translated into an enzyme, and together those enzymes make it possible to digest lactose. So it's really the presence of lactose that gets this whole thing rolling. So the repressor is listening for the presence of lactose. Once it's present, that makes a change in the repressor, causes transcription to happen. So an important question with these situations is, okay, we've had it off, we turned it on, how do you turn it off again? And this is a situation that I think people often expect there to be another signal. There's one signal for on and one signal for off. And it's really the beauty of this kind of system where you don't need another molecule. It turns out it's really just the absence of the first signal molecule that causes the signal to stop. So let's say we've done this where we transcribed and translated the lac operon genes. That's going to cause the lactose that's present in the cell and around the cell to be incorporated and digested into glucose and galactose. So as that happens, as long as there's lactose present, it's going to keep digesting the lactose that's there. Eventually, that's going to wear out all the lactose. It's going to be used up and turned into other forms of energy. So that absence of lactose means that this can't happen, means that this can't happen, means that this form of the repressor doesn't exist, and instead the form that is able to bind to DNA exists, and it's going to get in there and stop transcription. So it's really a question of how much lactose is there. So high amounts of lactose, transcription is on, low amounts of lactose, and that would turn things off. So very elegant, responds to the presence of the thing that needs to be digested. And when that thing is fully digested, then the need goes away, and so does the enzyme that would be required to change it. All right, I gave this away in a couple minutes ago, but maybe take a pause and remind yourself what's going on here. So this is showing the gene region being transcribed in the presence of the signal molecule. So the signal molecule is required to make this happen. The signal molecule is required to turn transcription on. That means this is inducible. So it's inducible means that it can be turned on, means that it's usually off. So this is a synonym for those other phrases I've been using, like usually off. So this is inducible. Okay, so in learning about the lac operon, they discovered mutants of basic gene regions that are part of this, and learning about the mutants can help us to understand the functions of the various molecules that are involved. So just want to walk through a few of these and show how a DNA mutation can give a certain kind of effect. So first kind we're going to look at is called LAC-I-. And so that is a defective gene, a mutated gene that makes a repressor that cannot repress. So just fundamentally unable to repress, it's unable to attach to the promoter. So regardless of whether there's lactose or no lactose, this cannot repress. There's never any repression, never any repression of transcription means transcription is always on. And they'd be able to detect that by the activity of this beta-galactosidase enzyme. So this is a LAC-I- mutant. This is a mutation in the gene for the repressor molecule. This is another kind of mutation, also at the LAC-I gene. When we talked about mutations, we talked about mutations that gain function or lose function. And so you can have a different effect depending on how you mutate the instructions for making a protein. And this is one that turns out to be a situation called a super repressor. And the super repressor mutant can attach to the DNA, but it's missing the ability to attach to lactose. And that's where the artist has decided to give us spikes on this enzyme, showing that it's just utterly insensitive to whether there's lactose or not. So similarly to the previous example, it doesn't matter if there's lactose or not, but different from the previous example, this one is always going to prevent transcription, whether there's lactose or not. So if there's lactose around here in the environment, it might normally turn off the activity of the repressor, but this mutant makes that impossible. It means that it's always on, always attached, I should say, to the promoter and keeps transcription from happening. So lactose present or lactose absent, no transcription happens here. And here one more example of a mutant. This is a mutant of the operator region, which again was a specific part of the promoter. And that's the region where the repressor would normally attach to. And this kind of mutant is a change in the recognition sequence of DNA so that a normal repressor molecule can't attach there. So yet another situation where it doesn't matter if there's lactose or not, the repressor cannot ever do its job of repressing. And therefore transcription is always on. And that's also related to the name there. This is called LAC-O superscript C. The C there, as I've shown you, stands for constitutive gene expression. So constitutive, otherwise what we've seen as always on, or the so-called housekeeping genes, are constitutive. This causes constant transcription of the operon because it has a mutant that prevents transcription from ever being turned off. All right, so we spent a lot of time on the lac operon. I think that's important to understand the process of that. That was an example where it was an inducible system under the control of a repressor. Now we have a repressible system. So again, what it means to be repressible means that it's usually on. So in the top example, we have transcription happening in the absence of a signal molecule. The bottom part of this repressor molecule is still open and ready. This is a different kind of repressor. This one can't repress unless it is attached to that signal molecule. So it's, again, the repressors would be specific to the genes that they act on, and this would be the repressor that's involved in controlling transcription of the TRIP operon. So the TRIP operon has 5 genes, and different genes encode different enzymes, and they're all involved in the process of converting a molecule into tryptophan. So that's where the TRIP, TRP abbreviation comes from. That's for tryptophan. And tryptophan is one of the amino acids. So making an amino acid is a process that needs to happen if there's any deficiency in tryptophan amino acid. So this is the flip side of what we saw for inducible systems. This is a repressible system where the cell is going to make tryptophan as long as there is a need for tryptophan. So as long as tryptophan, as I indicated once before, as long as this is drawn off to make proteins, then the cell is going to keep running low on tryptophan, and it's going to keep having the signal that causes transcription to happen. But if a situation happens where cell growth slows down, maybe the cell finds itself in an environment with abundant tryptophan, anything really where this abundance of tryptophan increases, if this builds up over time, then it doesn't make sense to keep making it, right? So that becomes a signal, this circle molecule of tryptophan, draw a circle all the way around, that becomes the signal molecule to interact with the repressor, and that causes the repressor to change shape and now be able to prevent transcription. So again, I think it helps to think about this kind of situation from a usefulness perspective. So it's useful to be able to turn off something that you're making if you've got too much of it. You can think of it as something that you're manufacturing. If you're manufacturing a certain kind of food, as long as people are buying it, it flies off the shelf, you're going to keep making that food. But if there are all the stores that have your food, have supplies on the shelves, it doesn't make sense to keep making that. You lose money, the cell loses energy to keep making this. So just like we saw in the lac operon also, it's only one molecule that is controlling expression. So it's only tryptophan. This molecule called chorismate has nothing to do with the signaling in this process. And if there's low tryptophan, that causes transcription to happen. If there's high tryptophan, that causes transcription to stop. So as you're reviewing these kinds of concepts, we've seen a few different ways to look at these. I think one thing that can be frustrating is that there are a lot of questions that are one answer or the other. So it's a bunch of 50-50 questions. These questions of the system being inducible or repressible. Is the gene usually on or usually off? Again, just to clarify that, usually when I use that word, I'm talking about in the absence of the signal molecule. And that's a substrate or product in the biochemical reaction controlled by the enzyme that's having the expression regulated. Activator or repressor. As we've seen, the definitions of activator and repressor have to do with what happens when they attach to the DNA. So an activator makes transcription happen when it's on DNA. The repressor prevents transcription when it's on DNA. It's kind of built into the names. It's pretty easy, I hope. And then what kind of signal molecule is interacting with the activator or repressor? What happens when the signal molecule attaches to the activator or repressor? So you can work through the examples we've done, and then also maybe come up with your own examples for, there's a variety of genes that are regulated in the same way in bacteria. So get some practice looking at those.